Rectus abdominis is a skeletal muscle. To locate rectus abdominis on abdominal wall of frog. To observe and understand, rectus abdominis is responsible for compression of abdominal organs and to flex the vertebral column in lumbar region. To understand that acetyl choline is responsible for contraction of rectus abdominis.
Rectus abdominis muscle of frog is a skeletal muscle. Skeletal muscle functions to move bones of the skeleton and thereby regulate movements of the body. Skeletal muscle tissues are also known as striated muscle. Skeletal muscle are voluntary type and are controlled by nerve cells of somatic system. Muscle tissues have special properties like excitability, contractility, extensibility, and elasticity. A physiological contact between somatic nerve cell and skeletal muscle is called neuromuscular junction. At this synaptic junction, acetyl choline is released from the end plate of somatic nerve cell. Acetyl choline is a natural neurotransmitter at neuromuscular junction, and after its release, it interacts with nicotinic receptors of skeletal muscle. This interaction is responsible for muscle contraction. Acetyl choline, after interacting with nicotinic receptor, induces change in membrane potential and induces influx of sodium ions and calcium ions inside the cell. This leads to muscle contraction.
Assembly setup. The assembly setup for experimentation of frog's rectus abdominis muscle preparation consists of organ bath. The organ bath consists of an outer jacket, which is also known as water bath, made up of steel or glass. Organ bath also consists of thermostatically controlled heating rod, which maintains uniform temperature of water in outer jacket. It also consists of tissue bath made up of glass with a capacity from 10 to 50 ml with both sides open. Isolated muscle preparation is mounted in upright position in this tissue bath. Aeration tube made up of glass has dual functions. One, it will aerate mounted muscle preparation, and second, it serves as the tissue holder. The other end of tube is connected to aerator. The bottom end of tissue bath is connected to rubber tubing, which serves as outlet. The bottom side of organ bath consists of an outlet for removing water from outer jacket. Recording lever. To record the responses of drugs on muscle preparation, one can use either simple lever or frontal writing lever. Frontal writing lever is made up of light aluminium and is designed in such a way that the writing tape rotates freely about its axis. For proper recording of responses, adjustment of magnification of lever is necessary. Magnification value is calculated by using following formula. Magnification value is equal to distance between fulcrum and writing point upon distance between fulcrum and the point of attachment to the tissue. For recording the effect on frog's rectus abdominis muscle, 5 to 10-fold magnification is required. For proper recording of responses and for proper relaxation of muscle preparation after drug effect, application of proper tension or load is required. For example, for frog's rectus abdominis muscle preparation, the tension prescribed is 1 to 5 gram. This tension is applied on longer arm of the lead. Sherrington's rotating drum. It is also known as kymograph. It consists of circular drum, speed regulating gear, shaft to which drum is connected, and main on or off switch. It has clutch lever, screw lift to alter the position of drum, and electrical striker. Kymograph paper is wrapped on circular drum and is smoked with black smoke of kerosene or benzene. To study the effect of acetyl choline on frog's rectus abdominis muscle, select healthy frog. Pit the frog with the help of pithing needle. Now, pin the limbs of the frog to dissecting tray. Remove the abdominal skin to expose rectus abdominis muscle. To isolate rectus abdominis muscle preparation, take side cut on rectus muscle. One can prepare 2 muscle preparation from a single frog. Now, tie a thread to the bottom and top of rectus muscle before removing muscle from the body of the frog. Mount this rectus muscle preparation in upright position in the organ path containing frog ringer solution under a tension of 1 to 5 gram. Now relax the tissue or muscle for about 45 minutes, during which wash this preparation with fresh quantity of ringer for at least 3 times. Now, this preparation is ready for experimentation. We have already learned the isolation technique of frog's rectus abdominis muscle and the setup of assembly. Now stabilize the mounted muscle preparation for 45 minutes in frog ringer solution. During this period, wash the muscle preparation with fresh quantity of frog ringer for at least 4 times. Start the drum after stabilization. Take baseline for about 30 seconds on smoked paper. Now stop the drum and inject 0.1 ml of acetyl choline solution in the reservoir containing mounted muscle preparation. Start the drum immediately. Record the contractions for 90 seconds. You can see upward movement of writing tip of frontal writing lever on smoked paper. Stop the drum. Now relax the contracted muscle by rapidly washing with fresh quantity of frog wringer. Because of this, writing tip of lever slowly reaches towards baseline. Repeat the procedure by using 0.2, 0.3 and 0.4 ml of acetyl choline solution. For recording of each response to increasing doses of acetyl choline, 30-second baseline, 90-second response time and total 5 minutes time cycle is to be followed. Now measure the height of each response in millimeter. Height of response is indicative of muscle contraction. You can see that with the increasing doses of acetylcholine, muscle contraction increases. Hence, we can say that cholinergics increase spasmodic action. You have learnt that acetylcholine shows nicotinic actions at neuromuscular junctions of skeletal muscle. The contraction of muscle is achieved by triggering muscle action potential. Acetylcholine also increases spasmodic action of rectus abdominis.